Today, please keep silent. Today we will covering two topics: intercellular junctions or connections and apoptosis. Both are important for short note from your point of view, examination point of view. Intercellular connections and apoptosis. दोनों एग्जाम्स में शॉर्ट नोट में आते हैं यूजुअली. So you know, as the name, they have to bind each other or to the basal lamina, to the basement membrane. For that there are different structures. We'll discuss those things here. So these intercellular connections, it connects from cell to cell and also cell to the basal lamina. Okay, that is the basal lamina is the part of the basement membrane. Okay, so there are two categories here: cellular junctions, cell to cell connections, and cell to basal lamina. Okay, cell to cell junctions में कौन आते हैं? Like zonula occludens, zonula occludens. Okay, जो occlude कर रहे हैं पूरा. Okay, then there are, इसको हम tight junctions भी कहेंगे, occlude कर रहे हैं तो. Then there are zonula adherens या gap junctions भी होते हैं. Gap junction is another form. Zonula occludens, that is tight junctions, then gap junctions, then zonula adherens. Zonula adherens. Okay. So, and there are another one that is desmosomes. ये क्या हो रहा है? Cell to cell connections के लिए ये होते हैं structure. Like zonula occludens, that is tight junctions. Then zonula adherens also, adhere connect. Then there are gap junctions. Then there are desmosomal junctions. Okay. Then the cell has to also join to the basement membrane. For that there are different structures like hemidesmosomes. Hemidesmosomes, they are connecting to the basal lamina. Okay. And also there are other focal adhesions, smaller adhesions. You can see in the structure, this is the basal lamina. You can see this is the part of the basement membrane. This is the epithelial structure, epithelial cell, and these are the, यहाँ पर क्या है? This is the basal lamina. Okay, ये तीनों मिलाकर basement membrane बनते हैं. Okay, then we will study each one by one. Coming on to the tight junctions, that is zonula occludens. You can see here in this structure, this is zonula occludens, the uppermost one. Okay, that is the tight junctions or zonula occludens. So the, इसमें क्या होता है? It obliterates the space between those two. There is tight junctions, the outer layer of the neighboring cells, they obliterate the space between them, forming a barrier to the transport. Obviously, it is also forming a barrier. Apart from the connecting cells, it is also forming a barrier to transport of solutes or solvents. Okay, so that the solutes and solvents, they cannot communicate from each other. Okay, इसके लिए होता है ये tight junctions. They ensure the impermeability by paracellular route. There are two routes by which any solutes or solvents can move. That is transcellular, cell को cross करते हुए जाता है, and there is paracellular also. Paracellular means दो cell के बीच में जो space होता है. Okay, that is a paracellular space. So these tight junctions ensure that those paracellular routes are also impermeable. Okay, these are found in structures like epithelium of the skin, endothelial cells of the blood-brain barrier, you know, endothelial cells, blood-brain barrier, and there are blood aqueous barrier also. So these are the examples where tight junctions are found. Then these are composed of different proteins. These tight junctions, इसमें जो junctions आ रहे हैं ना बीच में obliterate कर रहे हैं space. So there are different transmembrane proteins which are forming it. Those कौन-कौन से proteins होते हैं? This is occludin, claudin, and there are junctional adhesion molecule. Occludin, claudin, Then there are junctional adhesion molecules. You can see these are the proteins which are found. Occludin, then there are claudin, then there are junctional adhesion complex. Okay. So there, and you can see in the structure they are linked to the actin cytofilament. है ना, ये actin filament से connected होते हैं इधर दोनों cells. You can see here also these are the actin components, cytoskeleton components. These tight junctions are connected with them. These are they are connected linked to the actin cytoskeleton. Obviously, they are giving the structural support to the cell, integrity to the cell. Apart from that, it also helps in signal transduction. Transduction किसको कहते हैं? What is transduction? Transduction is change of one form of energy to another form of energy. Okay. So signal transduction into and out of the cell. सिग्नल को आपको क्रॉस करना, सिग्नल को प्रोसेसिंग में भी हेल्प कर रही है. So that is signal transduction into and out of the cell. So in this way, it is modulating the cellular proliferation, differentiation, and cellular behavior, obviously. Okay, depending upon the information. Then another type of junction, that is gap junctions. As the name itself suggests, there is gap in between those two cells. They are connected with each other through a gap junctions. Connections होता है उसमें. Protein is a connections. Okay. So they links the important feature is that it links the cytoplasm of the adjacent cells. This is the gap junction. ये क्या होता है, एक तरह से चैनल बना लेते हैं गैप जंक्शन. अंदर से साइटोप्लाज्म एक दूसरे से कम्युनिकेट कर सकते हैं एक दूसरे सेल से. ओके, दिस इज द इम्पोर्टेंट पार्ट दैट इट लिंक्स द साइटोप्लाज्म of the adjacent cells. Then it permits, obviously it will permit the exchange of ions, small nutrients also, and other messenger molecules, and it provides low impedance of electrical pathways. You will be studying later in nerve muscle physiology. There are synapses. Synapses क्या होते हैं? The junction between two neurons. So there are different types of synapses. There are chemical synapses, there are electrical synapses. So this provides a electrical synapse. This is an example of electrical synapse. Electrical synapse में gap junctions के through cross होता है मैसेज. Electrical synapse में होता है क्या, बेसिकली पासिंग ऑफ इंफॉर्मेशन. synapse के थ्रू क्या, एक न्यूरॉन दूसरे न्यूरॉन को पास करते हैं इंफॉर्मेशन. So there are different methods, like chemical messenger के थ्रू कर सकते हैं, and also through electrical method. That is called the electrical synapse. का एग्जाम्पल क्या होता है, गैप जंक्शन के थ्रू होते हैं वो, उसमें जो मैसेज इंफॉर्मेशन पास होता है. So through which electrical activity can be passed from one cell to another. So electrical synapse में जो मैसेज पास होता है, it is through the gap junction. Okay. Then we have discussed that these are formed of certain proteins. Those proteins are called connections. ये जो गैप जंक्शंस बन रहे हैं, एक दूसरे से कम्युनिकेट कर रहे हैं, इसमें प्रोटीन्स होते हैं which is called connections. These connections, actually they 6 connections come together from each side. दोनों साइड के सेल है ना, दोनों साइड से 6 connections come and form a bigger structure, and that is called connexon. Okay, connections 6 connections मिलेंगे तो connexon बनते हैं दोनों साइड. Then they join together with a central pore inside. This connection is a hemi-channel. दोनों साइड से hemi channel की तरह बना रहा है, which aligns with adjacent connections and they form the gap junction. In this way, this junction is formed. So the associated protein is connections. And you all must be knowing that gap junctions कहाँ पे मिलते हैं हमारे, हैं? हार्ट मसल्स में मिलते हैं ना, कार्डियक मसल्स में, एंड ऑल्सो इन स्मूथ मसल्स. स्मूथ मसल्स में आप पढ़ोगे GITs में, वहाँ पे सिंगल यूनिट होते हैं, मल्टी यूनिट होते हैं स्मूथ मसल. So single unit स्मूथ मसल is having those gap junctions. ओके, क्योंकि एक सेल से दूसरे सेल में इंफॉर्मेशन देना होता है, एंड दोज आर डन थ्रू दोज गैप जंक्शन in the cardiac muscle as well as in single unit smooth muscle cell. Okay, so this is the proteins which has been shown here. Connections, this one. Then there is zonula adherence. Zonula adhere कर रहा है एक दूसरे से. That is, this is having intermediate permeability. इसमें ऑक्लूडेंस में ऑलमोस्ट नेग्लिजिबल था, है ना. इसमें क्या है, दिस इज हैविंग इंटरमीडिएट परमेबिलिटी. गैप जंक्शन में परमेबिलिटी ज्यादा है क्योंकि साइटोप्लाज्म कम्युनिकेट कर रहे हैं. सो इट इज हैविंग इंटरमीडिएट परमेबिलिटी, एंड मेजर प्रोटीन इन्वॉल्व हियर इज कैडरिन एंड कैटेनाइन. प्रोटीन्स को याद रखना है, तो बिकॉज दीज आर स्पेसिफिक फॉर एवरी कनेक्शन. ओके, so major protein involved in zonular adherence is cadherin and catenine. Cadherin and catenines. You can see here. Cadherin and catenines. And this cadherin, ये जो cadherin जो होता है इसमें प्रोटीन, this is calcium dependent. Okay, calcium के इसका response depend करते हैं. And they are linked to actin cytofilament. एक्टिन कौन से होते हैं, ऑक्लूडेंस वाले भी एक्टिन से कनेक्टेड थे साइटोस्केलेटन. इट इज आल्सो कनेक्टेड विद एक्टिन साइटोस्केलेटन टुवर्ड्स द सेल, है ना. इस साइड में एक्टिन साइटोस्केलेटन हो. दे आर इन्वॉल्वड इन इंट्रासेलुलर सेल सिग्नलिंग अपार्ट फ्रॉम सेलुलर इंटीग्रिटी प्रोवाइडिंग सेलुलर स्ट्रेंथ. It is also involved in intracellular cell signaling and regulation of gene expression. Okay, cell में signal क्यों आता है? Obviously it has to influence the gene. Okay, nucleus में जो हमारे genes है, DNA, उसको influence करना होता है. Only then it can modulate the cells, है ना. cell को modulate तभी कर सकते हैं, differentiation कर सकते हैं. Okay, then next connection is the desmosomes. We have discussed about zonula occludens, gap junctions, then adherence. Then there is desmosomes. You can see here, this is the desmosome. है ना, ये इधर desmosomes दिखा रहा है. These are desmosomes, and these are also obviously located in the lateral surface of the cells. This is important that desmosomals are abundantly present in those tissues which are subjected to mechanical stress. है ना, जिसको ज्यादा स्ट्रेस झेलना होता है वहां पे ये ज्यादा होते हैं, है ना. स्पेशली इन स्किन, GI mucosa, myocardium and urinary bladder. And these are like the zonular adherence. Zonular adherence की तरह ही होता है, बट इसमें साइटोस्केलेटन जो होते हैं इट इज डिफरेंट. इट इज कनेक्टेड विथ इंटरमीडिएट फिलामेंट. वो क्या होते हैं, एक्टिन फिलामेंट से कनेक्टेड. जोनुलर एडेरेंस ये भी एडेरेंस की तरह ही होता है, बट द डिफरेंस इज इट इज लिंक टू द इंटरमीडिएट फिलामेंट, नॉट टू द एक्टिन फिलामेंट, है ना. साइटोस्केलेटन में हमने पढ़ा था ना, एक्टिन मायोफिलामेंट्स होते हैं, माइक्रोट्यूब्यूल्स, माइक्रोफिलामेंट्स, इंटरमीडिएट फिलामेंट. these are all forming the bones of your cell, cytoskeleton of the cell. Okay, so the difference is here it is linked to the intermediate filament. Then what is the protein involved in desmosomal connections? This is also related to cadherin superfamily, है ना. cadherin superfamily जोनुला adherens में भी था, but it is a different protein. The protein is different here. It is desmoglein and desmocolins. ना desmosome से उसी से नाम है, desmoglein and desmocolins. Okay, so these are the proteins involved in desmosomal connections. Then apart from this, obviously these are all providing the structural support. Apart from that, it is involved in other cellular signaling. and that is leading to the epithelial proliferation and differentiation. This is important that if there is mutation in these desmosomes, ये जो connections है desmosomal proteins में अगर mutation हो जाते हैं, it leads to certain diseases, especially of skin, है ना, especially of skin. उसमें क्या होता है, स्किन में स्पेशली डिजीज होता है दैट इज कॉल्ड पेम्फिगस, है ना. पेम्फिगस में क्या होता है, देयर इज अ, जो आपका लैमिनिन से जो कनेक्ट होता है ना, वो क्या होता है, वो फ्राइबल होने लगते हैं, सेल का कनेक्शन वीक होने लगता है ना, सेल का कनेक्शन वीक होने लगता है. वेरियस स्किन डिजीजेस हैव बीन एसोसिएटेड विद malfunctions of desmosomes like pemphigus, okay, which is an autoimmune disease, okay, and that autoimmune disease forms an autoantibody which destroys those structural support. desmosomes को प्रोटीन्स को डिस्ट्रॉय करते हैं. डिस्ट्रॉय करेगा तो क्या होगा, जो जुटा हुआ तो थोड़ा लूज हो जाएगा, isn't it? And there will be eruptions over your skin, okay. So there are other connections. This hemidesmosome was connecting the cell to the basal lamina, basement membrane, part of the basement membrane. So you can see in this structure, this is hemidesmosome, I think, hemidesmosome. This is hemidesmosome. It is connected. The hemidesmosome connection. It is having a protein called integrin. Integrin protein से connected होता है. Okay. And so it is a multi-protein complex that facilitates the stable adhesion of the basal epithelium. Epithelium को basal lamina से connect करेगा. So it will facilitate the stable adhesion to the underlying basement membrane. So these are important for stable adherence from epidermis to dermis. Epidermis और dermis के बीच में होता है. Okay. And the protein involved here is integrin, and this is important for differentiation and migration. Wound healing में important होता है hemidesmosome. Okay, hemidesmosome is important in wound healing also. So it is critical for cell differentiation and migration during wound healing. Then if there is dysfunction, disorder in this connection, hemidesmosome, तो क्या होगा, फ्रैजिलिटी, इट विल ऑल्सो कॉल फ्रैजिलिटी, जैसे उसमें भी हो रहा था लेकिन इसमें थोड़ा ज्यादा होता है, इंक्रीज फ्रैजिलिटी एंड ब्लिस्टरिंग ऑफ टिश्यू, बुल्लस इरप्शंस हो जाते हैं, है ना, बाहर की तरफ बुल्लस, इसलिए नाम है बुल्लस पेम्फिगॉइड होता है. यू विल स्टडी इन डर्मेटोलॉजी दैट देयर आर डिजीज कॉल्ड पेम्फिगस एंड देयर आर बुल्लस पेम्फिगॉइड. ओके, सीन पैथोलॉजिकली इन स्किन डिजीज लाइक बुल्लस पेम्फिगॉइड. ठीक है, अभी आपको कनेक्शंस पे ज्यादा ध्यान देना है, एग्जाम्स में तो वही रहते हैं, एग्जाम आपको एग्जाम्पल्स डालने होंगे. देन ऑल दिस प्रोटीन्स व्हिच वी हैव डिस्कस्ड, दिस कम अंडर सेल एडहेजन मॉलिक्यूल. उसको हम सब वो क्या कहते हैं, सेल एडहेजन मॉलिक्यूल है. दीज आर इंपॉर्टेंट सेल सरफेस प्रोटीन, सेल के सरफेस पे होते हैं, है ना. दीज आर इंपॉर्टेंट सेल सरफेस प्रोटीन मॉलिक्यूल्स व्हिच आर promoting the cell to cell and cell to basement membrane or matrix interaction. Okay, cytoskeleton interaction. So these are important for normal biological processes. ये किस चीज में काम आते हैं, हमारे adhesion molecule. These are important for embryonic cell migration, immune system, wound healing, है ना. These are involved in also in signaling pathways. Okay. Then there are different types of cell adhesion molecules. And cell adhesion molecule जो होते हैं वो different type के होते हैं. It expresses in 3 major domains. इसमें कौन कौन से डोमेन होते हैं, जो बाइंड करने वाला सेल एडहेजन मॉलिक्यूल है उसको 3 डोमेन होते हैं. एक बाहर की तरफ होगा, एक ट्रांससेलुलर जो सेल को क्रॉस कर रहा है, एक इनसाइड इंट्रासेलुलर कंपोनेंट. So extracellular domain and transcellular domain, then intracellular domain. Extracellular domain allows one cell adhesion molecule to bind to another on an adjacent cell, है ना. बाहर वाला क्या करेगा, बगल वाले से कनेक्ट करेगा ना, जो पास वाला होगा. So it helps in connecting one cell adhesion molecule to one cell to another cell, another adjacent cell. बीच में ये क्या है? This is a cell adhesion molecule. एक इधर जा रहा है, एक्स्ट्रा सेलुलर कंपोनेंट है दोनों का, तो एक दूसरे से कनेक्ट करने में हेल्प कर रहा है. बाइंडिंग डोमेन, दिस इज बाइंडिंग डोमेन. Then there is transmembrane domain. This is the transmembrane domain. जो ट्रांसमेम्ब्रेन पार्ट है ना, मेम्ब्रेन को क्रॉस कर रहा है. So the transmembrane domain links the cell adhesion molecule to the plasma membrane through the hydrophobic forces. Okay. Then there is cytoplasmic domain जो अंदर की तरफ है. It is directly connected to the cytoskeleton. अंदर में cytoskeleton होते हैं, filaments होते हैं, tubules होते हैं. So those domains are connecting with those cytoskeleton. Okay. Extracellular domain, then transcellular, then intracellular domain. This is connecting to this cytoskeleton. Then interactions between cell adhesion molecule can be mediated by एक तरह बाइंडिंग जो होते हैं वो होमोफिलिक बाइंडिंग भी होते हैं. होमोफिलिक मीन्स एक सेल का प्रोटीन जो है ना, एक सेल एडेशन मॉलिक्यूल में जो प्रोटीन है वो सेम तरह के प्रोटीन से बाइंड करेगा. ओके, लाइक कैडेरिन टू कैडेरिन. बाइंडिंग ऑफ वन एडेशन मॉलिक्यूल ऑन वन साइड टू द सेम एडेशन मॉलिक्यूल ऑन द अदर सेल. दूसरे सेल का जो सेम एडेशन मॉलिक्यूल उससे बाइंड जब कर रहे हैं, दैट इज कॉल्ड होमोफिलिक बाइंडिंग. कैडेरिन कैडेरिन लाइक. Then there is heterophilic binding, and it is binding to another protein, to the different type of cell adhesion molecule on a second cell. That is like selectin and mucin. That is called heterophilic binding. And there is a बीच में linker molecule होगा दोनों को जोड़ने के लिए, है ना? And that linker molecule is provided by laminin. Laminin भी हमारा extracellular matrix का part. बेसमेंट मेम्ब्रेन प्रोटीन. तो ठीक है. So these types of cell adhesion molecule, there are four broad categories of cell adhesion molecule. Cell adhesion molecule cell को adhere करने के लिए molecule है. So it is classified into four broad categories. That is integrins, which are binding through various receptors. Then there are immunoglobulin G superfamily. It belonging to this family are mainly immunoglobulins. They are mainly antibodies like ICAM, VCAM. यहाँ पढ़ोगे आगे जब blood vessels में पढ़ोगे clotting में, वहाँ पे आएगा ये. ICAM is intracellular adhesion molecule. VCAM is vascular cell adhesion molecule. Okay. Then PCAM, that is in protoplasm cell adhesion molecule, some adhesion molecule. Okay. So there are different adhesion molecules. So these all come under immunoglobulin G superfamily. Okay. Then there are cadherin superfamily. Cadherin superfamily क्या हुआ? It is calcium dependent. अभी हमने पढ़ा था cadherin proteins पढ़ा था. This is calcium dependent molecule that is mediating mainly the homophilic binding. And last one is selectin. These are lectin-like domains which are binding to the carbohydrates. So these have been classified into different types cell adhesion molecules. And what are the functions? Obviously they are connecting cell to cell. The cell is strengthened. Cell का strengthening करते हैं. They also play a role in cell movement due to the attachment of cytoskeleton. क्योंकि अंदर में क्या है, cytoskeleton से attached है. Like actin filament. Actin filament is involved in movement, isn't it? Then tubulin also there. Then transmit. It is also helping in signal transmission into and out of the cell. And in that way, it is influencing the genes involved. ना जीन के एक्सप्रेशन में हेल्प करता है, and it is involved in embryonic development and formation of different systems. It also prevents apoptosis. Do you know about apoptosis? Apoptosis क्या होता है? Programmed cell death. है ना? Our body does sometimes our bodies all the time they are doing apoptosis, because cells बहुत सारे क्या होते हैं सेनेसेंस में जाते हैं और वो डाई होते रहते हैं, नए सेल्स बनते रहते हैं, तो दे हैव टू बी प्रोग्राम्ड, दे हैव टू बी कंट्रोल्ड। वो जो कंट्रोल्ड सेल डेथ होता है दैट इज apoptosis, we'll discuss. This is also helpful in wound healing and inflammation, play a role in metastasis of the tumors. So there are different functions of cell adhesion molecules. This is all about intercellular connections. शॉर्ट नोट में आपको आएगा तो वो 4-5 टाइप्स के जो कनेक्शंस बताया आपको वो लिखने जरूरी है ना, दैट इज इंपॉर्टेंट, एंड द प्रोटीन्स इन्वॉल्व इन दैट, वही इंपॉर्टेंट है आपके लिए। सेल एडेशन मॉलिक्यूल ये पोस्ट ग्रेजुएशन में ज्यादा आते हैं, आपको पीजी एग्जाम्स वगैरह में एंट्रेंस में आ सकता है, ठीक है। देन कमिंग ऑन टू द एपोप्टोसिस, apoptosis as you can see in this figure, what is it showing off? It is showing off the falling of leaves. As you know, when there is autumn season, the trees shed off their leaves, like our body, when our cells are of no use or it becomes a threat to other cells, our cell programs the death of that cell, okay, that is called apoptosis. It is also called programmed cell death, okay. So it is a programmed cell death when the cells are no longer needed, or if it becomes a threat to the other cells, viability of other cells, then our cell undergoes a suicidal program, cell death. Suicide, it is a suicidal program. Obviously, our other cells are programming those death of the cells. That is called apoptosis. And there are different proteolytic enzymes which are involved here. आगे आप पढ़ेंगे mechanism of action में, वहाँ पे डिटेल में पढ़ेंगे. There are caspases. This is a very important enzyme, proteolytic enzyme, which is mandatory for apoptosis. What is the firstly what happens that those cells which are undergoing apoptosis, which have to undergo apoptosis, firstly it shrinks, पहले छोटे होते हैं. Then there is condensation of chromatin. Chromatin material जो होते हैं पहले न्यूक्लियर फ्रेगमेंटेशन होते हैं, डीएनए फ्रेगमेंटेशन देखेंगे डायग्राम्स में. Then there is chromatin condensation. Okay. Then there is disruption of cytoskeleton, and ultimately there are phagocytic cells nearby. And most important is the macrophages, big eaters, and who are small eaters. Body हमारा है ना, there are big eaters and small eaters. Macrophages are big eaters. Who are microphages? Neutrophils. Obviously, these are neutrophils. Neutrophils are micro eaters. So they will be activated and they will phagocytose those cells. Okay, those impending cells. So you can see there are two terms. You must be knowing necrosis and apoptosis. Necrosis क्या होगा? This is cell murder, cell injury, pathological. And apoptosis is cell suicide. So in दोनों में क्या difference you can see in the structure? These cells are swelling. ये swollen हो जाते हैं, वो shrinkage होता है cell. There is increase in cell volume in necrosis. They imbibe the surrounding fluid. Okay, and they increase in cell volume. There is cell shrinkage. Then there is loss of plasma membrane integrity. क्या होता है इसको जो प्लाज्मा मेम्ब्रेन, सेल मेम्ब्रेन जो चारों तरफ है, वो क्या होते हैं, वो डिस्ट्रॉय हो जाते है ना. डिस्ट्रॉय होने से क्या होगा, स्वेलन तो हो गया, जितना स्वेलन हो जाता तो वो डिस्ट्रॉय हो जाता. डिस्ट्रॉय होने के बाद क्या होगा, सेलुलर कंटेंट्स विल कम आउट, बाहर आ जाएंगे. जब बाहर आ जाएंगे तो क्या होंगे, सेल के अंदर में बहुत सारे इंफ्लेमेटरी सेल मेडिएटर्स होते, वो क्या होंगे, वो बाहर आ जाएंगे, है ना, साइटोकाइंस एंड ऑल दैट, एंड इट विल प्रोड्यूस इन्फ्लेमेशन. क्योंकि अंदर में जो सेल्स है, सेलुलर कंटेंट है, वो बाहर आ जाने से क्या होता है, इट इंड्यूसेस इन्फ्लेमेशन. इसमें क्या है, there is plasma membrane blebbing, but the membrane is, टूटते तो हैं, but they're in form of apoptotic bodies, है ना, अलग अलग क्या होते हैं, इस तरह से बॉडीज बनते हैं, इस तरह से अलग अलग शेड ऑफ हो जाते हैं, बट द मेम्ब्रेन इंटीग्रिटी इज मेंटेन्ड so that इसके जो कंटेंट है वो बाहर नहीं आते, so it is not associated with inflammation, उसमें inflammation नहीं होता है ना. So leakage of cellular contents in necrosis, but इसमें क्या है, formation of apoptotic bodies with the integrity of the membrane. It is called death by injury and it is called death by suicide, isn't it? This is the steps which are involved in apoptosis and necrosis. You can see the normal cell, when those cells who are undergoing apoptosis, who has to go and apoptosis, programmed cell death, what they do, firstly there is cell shrinkage and there is nuclear fragmentation and DNA fragmentation within it. Nuclear and DNA fragmentation. Fragment होने के बाद क्या होता है? There is chromatin condensation. This is important point. Chromatin condense हो जाते हैं. Fragment होने के बाद chromatin gets condensed, है ना? And what happens that it forms bodies. It sheds off its bodies. That is called apoptotic bodies. जैसे क्या होता है? आपने पढ़ा होगा तीनों साइटोसिस अंदर में आ जाता है, फैगोसाइटोसिस, एक्सोसाइटोसिस, है ना, बाहर जाते हैं, उसी तरह से ये पिंच ऑफ हो जाते हैं. ओके, एंड देयर आर फॉर्मेशन ऑफ एपोप्टोटिक बॉडीज, बट द ऑर्गेनल्स आर स्टिल फंक्शनल, दे आर हैविंग द ऑर्गेनल्स विद इन इट. ओके, एंड अल्टीमेटली दोज आर फैगोसाइटोज्ड. These apoptotic bodies are phagocytosed by those macrophages. Whereas in necrosis, you can see there are cellular swelling. Shrinkage होता है, इसमें swelling है ना. There are also formation of blebs, but what happens that its membrane gets disintegrated and all the cellular contents come out. Breakdown of plasma membrane, the organelles and nucleus all are broken down and then come out. And that's why it is associated with inflammation. So apoptosis is not associated with inflammation. Why is it so? Why is it so? Because the cellular contents, the organelles are retained within the membrane, है ना, अंदर के contents बाहर नहीं आ रहे, बाहर आएंगे तो वो inflammation कराएंगे. The organelles are retained within their plasma membrane and those apoptotic bodies are rapidly phagocytosed without the release of any cytokines, inflammatory mediators. And this phagocytosis is facilitated by certain cell surface proteins. आगे देखेंगे mechanism में, वहाँ पे और ज़्यादा clear होगा. Phagocytosis is achieved rapidly with little damage to the rest of the tissues. So what are the changes in apoptosis? There are pathophysiological changes and there are biochemical changes. What are pathophysiological changes? This apoptosis may involve a single cell or a cluster of cells. What are pathophysiological changes? This is the earliest feature. The cell starts shrinking. Okay, apoptosis में क्या होता है? पहले shrinkage होना शुरू हो जाता है cell का. Then the cytoplasm जो अंदर में cytoplasm, it becomes eosinophilic and the organelles are remaining normal. Organelle disrupt नहीं होते, उसके membrane intact होते हैं. Okay, the cytoplasm becomes less in number, that's why there is shrinkage. Cytoplasm क्या होगा? कम होने लगेगा, scanty होने लगेगा, but and it becomes eosinophilic and the organelles remain normal. Then there is nuclear, as we have discussed, nuclear fragmentation and DNA fragmentation. After that, there is chromatin condensation. Chromatin condense होते हैं. There are different genes which are involved in all these things. Okay, this is chromatin condensation is a characteristic feature. It is important that chromatin condensation is the characteristic feature. Then there is formation of cytoplasmic waves and apoptotic bodies, and then ultimately it is getting phagocytosis. You can see chromatin condensation, the apoptotic bodies, okay. Then there is phagocytosis. Then there is biochemical changes also. What is the biochemical changes like? There is cytoskeletal damage. Cytoskeletal जो प्रोटीन होते हैं अंदर में वो तो डैमेज होगा. So there is proteolysis. उसके associated जो प्रोटीन्स है उसका लाइसिस होगा. Then there is nuclear fragmentation by endonucleases. है ना, endonuclease enzyme होते हैं apart from caspases. So that it is fragmenting. Then this is important point. That is this glycoprotein molecule phagocytosis जो है ना, phagocytosis कौन कर रहा है? मैक्रोफेजेस कर रहे हैं apoptotic bodies को. तो apoptotic bodies पे there are certain signals which are present. जो apoptotic bodies होते हैं, जो cells die करने वाले, apoptosis होने वाला है जिसमें, उसके cell surface पे there are certain signals which comes over it, and जिसके चलते क्या होता है, मैक्रोफेजेस उधर अट्रैक्ट हो जाते हैं, and those are thrombospondin. No, this is glycoprotein, that is thrombospondin, and this is glycolipid, that is आपने पढ़ा था phosphatidyl serine and phosphatidyl ethanolamine. नहीं पढ़ा था ना? कहाँ पे पढ़ा था? Where? Cell membrane के structure में there are phospholipids. There are different types of phospholipids. Those plasma membrane were asymmetrical. अंदर वाले अलग थे, बाहर वाले कुछ अलग थे. तो ये phosphatidyl serine and phosphatidyl ethanolamine was inside the cell membrane. अंदर वाले जो leaflets में थे. तो ये क्या होता है? ये बाहर की तरफ चले जाते हैं. Cell membrane होते हैं अंदर में, अंदर वाले cell membrane पे. So what happened that those apoptotic bodies, उसके cell membrane तो है उसमें भी, उसमें ये जो glycolipids है वो cell surface के ऊपर आ जाते हैं, and these act as a attractant for those macrophages. So this facilitates the recognition by macrophage. Macrophage उसको पहचान लेते. So I have to indulge this, okay, for phagocytosis. This term was first introduced by these scientists, okay. It occurs, this apoptosis occurs in different physiological condition as well and in pathological condition as well. है ना? Small some pathological condition as well. On physiological conditions में होते हैं apoptosis physiologically. As you know, during the formation, during the embryogenesis, what happens that your fingers, they are all joined together by wave. Wave से जुटे होते हैं सारे, है ना? ये क्या होता है? इसका स्लफिंग होते हैं in between the fingers, है ना, वो tissues का, and that is due to the apoptosis. Okay, so during the formation of the fingers and toes of the fetus, this apoptosis, it removes those wave tissues in between the fingers and toes. And during the sexual development also, you must be knowing there are Wolffian duct and there are Mullerian duct, है ना? So one has to regress. ना Mullerian duct में females males में क्या होता है? Wolffian duct, है ना? और opposite to that. So one has to regress. So this occurs through those apoptosis. So this is another physiological example. Then there is during the time of menstruation, there is cyclical breakdown of endometrium, and there is withdrawal of progesterone. Withdrawal होता है ना, no, hormones का, that leads to the menstruation, which is also due to the apoptosis. This breakdown of endometrium. है ना? There is involution of thymus in the early age, है ना? It becomes involuted. Okay. Then there is pathological apoptosis also. ये तो physiologically है. Pathological क्या है? Like those cells which are infected with virus, उसको भी क्या होता है? Apoptosis. उसको भी क्या करना है? Cell को death करना है. So it undergoes apoptosis. Those cells which are infected with certain viruses, which may be harmful for your body. So apoptotic mechanism are stimulated, and there are abnormalities of apoptosis also. If this apoptosis mechanism is not in line, it may lead to certain degenerative disorders like Alzheimer's disease, Parkinson's disease, है ना. ये apoptosis का क्या होता है, abnormalities होते हैं. Then there is depletion of CD4 T cell count in AIDS. ये भी pathological apoptosis के चलते होते हैं. Then apoptosis of cancer cells which is mediated by chemotherapy. हम chemotherapy देते हैं तो क्या करते हैं, apoptosis stimulate होता है, and those वो क्या करते हैं, हमारे कैंसर सेल्स जो थे उसको कम करते हैं, डिस्ट्रॉय करते हैं. Then what is the mechanism involved here? There are two important enzymes which are involved. That is caspases. है ना, इनिशियली बताया था आपको, दिस इज प्रोटेलिटिक एंजाइम. कैस्पासेज का फुल फॉर्म क्या है, सी से क्या हुआ, सिस्टीन. देन ए से एस्पार्टेट. ओके, एस्पार्टिक एसिड एंजाइम, सिस्टीन एस्पार्टिक एसिड एंजाइम. This is the proteolytic enzyme. This is very important, so that the apoptosis mechanism is stimulated. And there are two types of caspases. There are initiator, है ना, इनिशिएटर है, एग्जीक्यूशनर है. एक इनिशिएट कर रहा है, एक एग्जीक्यूट कर रहा है. कैस्पेसेस में जो 8, 9, 10 कैस्पेसेस होते हैं वो इनिशिएटर होते हैं, एंड 3, 6, 7 इट इज द एग्जीक्यूशनर कैस्पेसेस. देयर आर डिफरेंट टाइप्स ऑफ कैस्पेसेस. Okay. So, and there is another important enzyme which is involved in apoptosis is endonuclease. That is this is causing the nuclear fragmentation and DNA fragmentation. ये mechanism पढ़ा था हमने. So there are different pathways, intrinsic pathway and there is extrinsic pathway, है ना. apoptotic pathways जो mechanism हो रहा है, दो mechanisms हो रहे हैं. एक सेल के अंदर स्टिमुलेट हो रहा, इंट्रिंसिक mitochondrial pathway भी कहते हैं, क्योंकि mitochondria is mainly involved in those pathways. Are you able to understand me or not? Please tell me. Are you able to get to me or not? समझ में आ रहा है? So extrinsic pathway and intrinsic pathway. There are extrinsic pathway and there are another pathway that is called porphyrin or granzyme pathway. Factors involved in apoptosis. There are pro-apoptotic factors. There are certain factors which has to be stimulated. Like apoptosis होना है तो ये factor ज़्यादा stimulate हो जाएगा. BAC and P53. ये पैथोलॉजी में भी apoptosis आपको पढ़ना होता है, सेकेंड ईयर में पढ़ोगे. And there are anti-apoptotic factor also, which is preventing the apoptosis of normal cells. This is BCL2, BCLX, and MCL1. है ना, pro-apoptotic, BAC and P53. There are anti-apoptotic, that is BCL2. Then there are arbiters. Arbiters क्या हुआ, बैलेंस बना के रखते हैं, pro-apoptotic और anti-apoptotic बैलेंस बना के रखते हैं, है ना. That is beam, bag, HID and tumor. Okay, so these are different factors which are involved in apoptosis. Pro-apoptotic, anti-apoptotic, and there are balancers. So this is the mechanism actually. This is intrinsic mechanism which is mediated through mitochondria. Mitochondrial pathway कहते हैं. Then there is extrinsic mechanism, वो बाहर से cell surface protein के through आता है. What happened that होता क्या ultimately, intrinsic mechanism is the most often pathway. 90% apoptosis is pathway, intrinsic pathway, and rest is through the extrinsic. And okay. So how it is happening? What happens that in the mitochondria, initially if there is certain stimulus, internal stimuli, if there is certain toxins, hypoxia, radiation, what happens that these radiations, toxins or hypoxia, this is stimulating the arbiters. जो arbiters में जो balancers है उसको stimulate करते हैं. वो stimulate होने से क्या होता है, those arbiters they shift pro-apoptotic factors को ज़्यादा stimulate करते हैं. Okay, and pro-apoptotic factor को ज़्यादा stimulate कर देते हैं. Okay, so what happens that in the mitochondria के अंदर में क्या होता है, inner and outer membrane होता है, you know, and the inner membrane, जो अंदर वाला जो membrane हो, matrix जो है, this is having the enzyme that is called the cytochrome C. This cytochrome C whenever it is inside the matrix, तो apoptosis induce नहीं होगा. Okay, if anyhow this cytochrome C ये अगर बाहर आ जाता है ना, मेम्ब्रेन से बाहर आ जाता है, what happens that it triggers apoptotic factors like caspase. Okay, and in the outer membrane, ये तो अंदर में था, अंदर आउटर मेम्ब्रेन में क्या होता है, देयर आर एंटी एपोप्टोटिक फैक्टर्स. एंटी एपोप्टोटिक कौन से थे? एंटी एपोप्टोटिक फैक्टर BCL2 BCLX है ना. हाँ, ये एंटी एपोप्टोटिक थे, ये आउटर मेम्ब्रेन पे होते हैं. और वो क्या करते हैं, एंटी एपोप्टोटिक होने के चलते क्या होता है, जो साइटोक्रोम सी है उसको अंदर ही रखता है वो. ओके, इसका परमेबिलिटी को मेंटेन करके रखता है. ओके, अंदर नहीं आने, बाहर नहीं आने देता. जैसे ट्रिगर्स स्टिमुलेट होते हैं थ्रू द आर्बिटर्स, रेडिएशन हुआ क्या इससे हुआ, तो वो क्या करते हैं, व्हाट हैपेंस दैट, व्हाट इज हैपनिंग, दोज प्रो एपोप्टोटिक फैक्टर्स इन्फ्लुएंस हो जाते हैं, एंड दैट इज मेकिंग अ चैनल. वो क्या करते हैं, जो मैट्रिक्स है और मेम्ब्रेन है वो एक चैनल बना लेते हैं. थ्रू दैट व्हाट हैपेंस दैट, जो एंटी एपोप्टोटिक फैक्टर को इनहिबिट हो जाता है ना, और प्रो एपोप्टोटिक फैक्टर स्टिमुलेट हो जाता है. So it becomes a channel and cytochrome C comes out from the matrix, and that will stimulate those caspases. बाहर में caspases होते हैं ना, initially pro-caspases are there, that is getting stimulated and becomes caspases, active caspases. So caspases 9 है ना, caspases 9 था, this was initiator. executioner caspases था, एक initiator थे. So these initiator caspases are stimulated, caspases 9. Okay, and ultimately it will stimulate the executioner caspases 3, 6, 7. Okay, this is the intrinsic pathway, and there is extrinsic pathway also. There are cell surface protein. Cell surface के ऊपर में क्या होते हैं, there are proteins and they are having ligands or fast proteins. then tumor necrosis factor protein, उसके लिए ligands होते हैं, cell surface proteins होते हैं, receptor होते हैं, and these are called death receptors. ये जो है ना, FAS and TNF and P50, they are called death receptors. जैसे ही स्टिमुलेट हो जाता है FAS और TNF, ये रिसेप्टर पे सेल सरफेस के ऊपर में इसके लिए रिसेप्टर होते हैं, जैसे इससे आके बाइंड करते हैं. ओके, व्हाट हैपेंस दैट दिस रिसेप्टर्स आर स्टिमुलेटेड, एंड अंदर में क्या होते हैं, देयर आर FAS activated death receptors, death domain, fast activated domain. This is getting activated, and ultimately it activates the caspase 8. These are also initiators, है ना, initiators है ना. So ultimately they will all stimulate the executioner caspase 3, 6, 7. जैसे ये executioner caspase activate होता है, there it causes DNA fragmentation, cytoplasmic microchromatin को condensation होगा, there are all the mechanisms of the apoptosis will be triggered. Okay, and there are perforins, अभी आपको बताया था, एक और था पाथवे, उसके लिए क्या था, वो क्या करते, caspase 10 के थ्रू काम करते, it perforates the cell membrane. ठीक है, so ultimately what will happen, there will be cell breakage of the cell death will occur, and you know जैसे apoptotic bodies आएंगे उसके ऊपर में क्या होगा, जो ग्लाइकोप्रोटीन जो सरफेस पे आ जाएगा, जो नियर बाई माइक्रोफेजेस है वो उधर अट्रैक्ट होकर आ जाएगा, एंड इट विल इंगल्फ दोस एपोप्टोटिक बॉडीज. सो इन दिस वे दिस मैकेनिज्म इज occurring. This is all the same thing which we have discussed in the diagram. That is the intrinsic pathway, that is the mitochondrial pathway. Attendance we have not started now. We started fast. So the intrinsic pathway, that is the mitochondrial pathway, it is the result of, as we have discussed, result of virus stimulus like radiation, hypoxia, toxins, hypothermia. It initiates a series of intracellular events leading to the cell death. है ना, इसमें ज्यादा डिटेल में नहीं लिखा है, डायग्राम में it was much more clear. Then there is, they do not act through the extrinsic pathway, is acting through these death receptors, FAS and TNF receptor gene family. इसको हम death receptor कहते हैं, and it is stimulating this formation of death inducing signaling complex, that is also called death domain, है ना. fast activated death domain. Then there is perforin or granzyme pathway. It is acting through caspase 10. It is actually, it relates to the death of tumor cells. वायरस इन्फेक्टेड जो सेल्स है उसको एपोप्टोसिस होना है, तो इसके थ्रू होता है, परफोरिन के थ्रू परफोरेट कर देते हैं उस सेल, है ना, परफोरेट and granzyme pathway. Perforin involves the release of cytoplasmic granules through a pore into the target. So both intrinsic and extrinsic, it is acting through the executioner caspases 367, and these amplify the apoptotic process leading to the cell death. Then obviously there is elimination is through the macrophages. Macrophages have to identify those apoptotic bodies which have to be cleared off, and this is the molecule which is there, thrombospondin. Apart from this, phosphatidylserine and phosphatidylethanolamine, ज्यादातर ये इनसाइड में होते हैं सेल मेम्ब्रेन के, है ना, बाइलेयर स्ट्रक्चर होता है ना, जो अंदर वाले लेयर पे होती है, ये बाहर चले आते हैं. And this is all about the apoptosis. Another method that is called autophagy भी होते हैं sometimes. It is a form of cell death where the cell sequesters its cytoplasm and organelles in vesicles, and ultimately it is acting upon by the lysosome. So it is having a cannibalization effect. It is not dependent on caspases. And necrosis we have discussed. Few questions. Cellular junctions present in cardiac muscles. Gap junctions नहीं होंगे ना cardiac muscles में, all except है ये. They are different. This is all except. Gap junctions के अलावा भी होते हैं, हाँ? No, zonula occludens, है ना, occludens नहीं होते उसमें, ठीक है. Adherents भी होते हैं small amount में, macular adherents. Then there is exchange of cytoplasmic molecules, gap junctions. True about gap junctions or all except, you can tell, I think. Can answer this question? Huh? Last one. Space कम होता है. It is not so much. It is approximately 2 to 3 nanometer only. That's all.